Hello and welcome to the Arise interview. 60 minutes of big questions about the big stories from the news and beyond, with fresh insight and critical analysis. I'm Charles Anyagolo. Coming up in the next hour, a major scientific breakthrough using artificial intelligence. Scientists in the US have discovered a powerful antibiotic that kills some of the most dangerous drug-resistant bacteria in the world. The discovery has been made by a group of researchers from the Massachusetts Institute of Technology. So, are we on the verge of a new age of antibiotic drugs discovery? We'll speak to one of the leading pharmaceutical researchers in West Africa. And later, as the number of cases and the number of deaths linked to the coronavirus escalate around the world, with cases now in 31 countries, there are growing calls from conservationists for a permanent ban on the wildlife trade in China. The outbreak is believed to have started at a wildlife market in Wuhan. We'll speak to the head of a conservation foundation in Nigeria that's joined others around the world calling for the wildlife ban. Coming up in a moment. Now, as we mentioned at the outset, researchers at the Massachusetts Institute of Technology, or MIT in the US, have used artificial intelligence to develop a new antibiotic compound that can kill some of the most antibiotic-resistant strains of bacteria. They created a computer model of millions of chemical compounds and used a machine learning algorithm to pick out those which could be effective antibiotics. They then selected one particular compound for testing and found it to be effective against E. coli and other bacteria. Most new antibiotics developed today are variations on existing drugs using the same mechanisms. The new AI antibiotic, though, uses a different mechanism than these existing drugs, meaning that it can treat infections that current drugs cannot. Well, in a moment, we'll gauge the significance of this scientific breakthrough. But first, what causes antibiotic resistance in the first place, and why is this happening? Well, in this online animated video, Kevin Wu details the evolution of this problem.
What if I told you there were trillions of tiny bacteria all around you? It's true. Microorganisms called bacteria were some of the first life forms to appear on Earth. Though they consist of only a single cell, their total biomass is greater than that of all plants and animals combined. And they live virtually everywhere, on the ground, in the water, on your kitchen table, on your skin, even inside you. Don't reach for the panic button just yet. Although you have 10 times more bacterial cells inside you than your body has human cells, many of these bacteria are harmless or even beneficial, helping digestion and immunity. But there are a few bad apples that can cause harmful infections, from minor inconveniences to deadly epidemics. Fortunately, there are amazing medicines designed to fight bacterial infections. Synthesized from chemicals or occurring naturally in things like mold, these antibiotics kill or neutralize bacteria by interrupting cell wall synthesis or interfering with vital processes like protein synthesis, all while leaving human cells unharmed. The deployment of antibiotics over the course of the 20th century has rendered many previously dangerous diseases easily treatable. But today, more and more of our antibiotics are becoming less effective. Did something go wrong to make them stop working? The problem is not with the antibiotics, but the bacteria they were made to fight. And the reason lies in Darwin's theory of natural selection. Just like any other organisms, individual bacteria can undergo random mutations. Many of these mutations are harmful or useless, but every now and then, one comes along that gives its organism an edge in survival. And for a bacterium, a mutation making it resistant to a certain antibiotic gives quite the edge. As the non-resistant bacteria are killed off, which happens especially quickly in antibiotic-rich environments like hospitals, there is more room and resources for the resistant ones to thrive, passing along only the mutated genes that help them do so. Reproduction isn't the only way to do this. Some can release their DNA upon death to be picked up by other bacteria, while others use a method called conjugation, connecting through pili to share their genes. Over time, the resistant genes proliferate, creating entire strains of resistant superbacteria.
Extraordinary. Well, for more on this, I'm joined now in the studio by the Director General of the National Institute for Pharmaceutical Research in Nigeria, Dr. Obi Adigwe. Thank you very much indeed for coming in.
Thank you for having me, Charles.
So basically, they've been able to harness the power of artificial intelligence to address the antibiotic resistance crisis that we just saw in that little video. Tell us how they did it.
So the use of artificial intelligence in drug discovery has been on for just over a decade now. Previously, it used to be the traditional in vitro and in vivo tests. In vitro meaning in the test tubes, and in vivo meaning in animal models. But over a decade ago, people started to understand the ability for you to use artificial intelligence to create models which could quickly screen some of these synergies, products which have activity and what they have activity against. But what makes this particular discovery quite important is that prior to now, the type of machine learning, the type of AI that has been used has been mostly supervised. And there is a more unsupervised approach this time around, which is sort of like telling the artificial intelligence not to follow a definite pattern, but to scan through everything and then find us some new intervention.
That's fascinating.
It is fascinating.
I mean, that is absolutely fascinating.
Yeah, and it is. I think this particular discovery is actually going to change the paradigm in drug discovery. So when we were in pharmacy school 20 years ago, we used to learn that first you'd get phytomedicines or the chemical molecules, then you test them in test tubes, and then afterwards, if they have activity against a particular microorganism, then you start testing in models. This takes hundreds of hours and millions of dollars. And what these guys have done is develop an artificial intelligence model that runs 100 million of these combinations in hours.
So let me just get this, because you're a scientist. I'm not. And a lot of people out there are not. So essentially, they used data to train a computer to learn molecular features associated with antibacterial activity. They then applied that learn model, if you like, to a drug repurposing library and challenged the model to identify molecules that are predicted to have antibacterial activity, but don't look like existing antibiotics. In other words, they challenge them, don't look for the ones that exist, look for new ones. And one molecule out of that library fit those criteria.
So the slight correction in what you said, they didn't say don't look for the ones that look like antibiotics. They just said look for anything.
Right.
Look for anything.
So when they then find everything, they can pick the ones. Brilliant.
Absolutely.
Yes.
And that's what makes AI different from a scientist.
Right.
I've been trained, pharmacy school, science training, medical school, you're talking of decades of training. And in the decades of training, in addition, in as much as you're being trained to understand the technical aspects of science, observation, comparison, instinctively, there are certain, how would I put it now, sensibilities you pick up along the way.
Sure.
You become prejudiced to certain preconditions. So you know that a benzene model is key to a particular compound.
Yes.
So if you see 1,000 formulas or 1,000 structures of different models, instinctively the scientist will move to the structure that looks like what he learned in school.
That is
AI doesn't have
those
that.
prejudices.
No
basically.
prejudices.
Right.
That was the word I was looking for.
Yes.
AI has no prejudices. And that's what makes this particular find interesting.
And so going to that particular find, that molecule that was then isolated turned out to be halicin.
Halicin, yes.
Right, one of the most powerful antibiotics discovered today. What was their knowledge of halicin before they went through the process that you've just described? I understand it was virtually zero, basically.
Well, it had some activity against diabetes.
Right.
So that's how it found its way into that database that you spoke of earlier on. The good thing about a lot of research institutes in the developed world is that they maintain a huge database.
Yes.
So halicin had been tried in the past and it was found to have some diabetic activity, but for some reason it didn't move from the preclinical stage to the clinical stage.
Right.
And so it found itself in that database, and you have so many others in that database. So what they did was, when they reconfigured the assignment for the algorithm,
right,
that brought this up, they looked at the database, but this time around repurposed it for antimicrobial activity.
Right.
And then it popped up.
So this is a case, just trying to follow your very intelligent description there. This is a case of something being right under their noses.
Absolutely.
And AI being able to tell them to look at this in a different way.
Absolutely.
So is it fair to say that they would never have looked at it in a different way had it not been for AI? We've got about 30 seconds before we have to take a break.
Never is a strong word. As scientists, we don't say never, but almost never.
Right.
Almost never. And I think what makes this particular finding interesting in the science world is that halicin is a totally new group of antibiotics. It's not a penicillin, it's not a macrolide, it's not a cephalosporin, it's entirely new.
Well, okay, just hold that thought. We're going to come back to you in a moment. You're watching the Arise interview, plenty more still ahead as we continue our chat about what could be a new age of antibiotic drug discovery using artificial intelligence. Stay with us. Welcome back to the Arise interview. I'm Charles Aniagolu, and as you've been hearing, scientists at MIT in the US have developed a new platform that allows them to harness the power of artificial intelligence to usher in a new antibiotic drug discovery. Their approach has revealed an amazing molecule that is arguably one of the more powerful antibiotics that's ever been discovered. This new approach to drug development could make identifying different drug compounds to which bacteria are resistant faster and considerably less expensive than other approaches. The researchers trained their computer model on 2,500 existing molecules and then tested it on a library of 6,000 compounds to identify molecules which could kill E. coli bacteria. The model identified one particular molecule as a potential target, and when the researchers tested it in the laboratory, they found it could kill treatment-resistant bacteria. Here are scientists at the Broad Institute talking about how they're helping to address the challenge of antibiotic resistance.
Antibiotics were first discovered and deployed more than 75 years ago, but the development of new antibiotics largely stalled by the 1980s. In the last 30 years, a disturbing trend has emerged.
According to the World Health Organization, more than a quarter of healthcare-associated infections in long-term acute settings exhibit some form of antibiotic resistance.
This means that with antibiotic resistance on the rise, the antibiotics that we've relied on for almost a century are becoming less effective. One solution is to develop new antibiotics. Here at the Blaney Lab at the Broad Institute, we believe that an alternative solution is to combine antibiotics with existing drugs.
In particular, gram-negative bacteria like E. coli feature a thick outer membrane that keep antibiotics from getting in and effectively treating an infection. Recent studies have shown that combining antibiotics with other existing drugs can give those antibiotics a fighting chance.
One example is Loperamide, a drug commonly used to treat diarrhea, but that has also shown an ability to disrupt gram-negative cell envelopes.
There are over 2,000 FDA-approved drugs used in hospitals and clinics today. And so to test every single pair of these drugs against just a single bacteria would take millions of tests and would require complex and expensive robotic equipment.
We wanted to make this easier, so we designed this microwell array device that effectively tests about 1,000 pairs of antibiotics and other drugs.
So what we do is we make these tiny droplets that each contain bacteria as well as a single antibiotic or a single drug. And then we load pairs of these droplets into each microwell. We color code each droplet to identify the specific antibiotic drug pair that got loaded into each microwell. And then we can use microscopy to actually track the growth of the bacteria within each microwell. And what we're looking for are instances where the bacteria couldn't grow. And in these cases, it might mean that we've discovered a synergy between the drug and the antibiotic.
For our first full-scale test, we screened 10 antibiotics against more than 4,000 compounds from the Broad Institute's drug repurposing library. This library is a collection of compounds that are in development or clinical use for other diseases. After just 10 days of testing against E. coli, we discovered more than 10 promising combinations for further study.
We think that with time, we'll be able to scale up this technology so that many labs can screen 10 times as many drug combinations at much lower costs than an industrial screening facility. And our lab and collaborators are working to adapt this technology to a wide range of disease areas in addition to infectious disease.
Very educational. And with me in the studio, helping us analyze the significance of the scientific breakthrough, the Director General of the National Institute for Pharmaceutical Research in Nigeria, Dr. Obi Adigwe. Thank you for staying with us. We're talking about halicin before we went on a break. Clearly a molecule that can find antibiotic-resistant pathogens. They've been looking at E. coli, as we heard in that video, first of all. What else can it possibly be used for? Because potentially it can have activity against a broad range of pathogens, which we desperately need antibiotics for.
Yeah, so the study looked at activity of halicin against 36 different types of bacteria. Majority of which had a profile of, antibiotic resistance. And like you rightly mentioned, E. coli, Escherichia coli was one of them. But very interestingly, it also was found to have activity against things like Clostridium difficile. Now, Clostridium difficile has plagued the Western world and possibly the developing world. Is just that we don't have the data to support that, but has plagued the Western world for ages. Clostridium difficile and methicillin-resistant Staphylococcus aureus. These two organisms have been the bane of hospital stay in the West. So Clostridium difficile is usually found in your gut normally, but if you've had issues, it could go on to cause life-threatening colitis.
Right.
Another particular product which is of interest that it was found to have activity against was Acinetobacter Bomani. Again, this is one of the, I think on one of the sites, I can't remember if it's CDC or the other site in the UK, it's listed as one of the most dangerous bacteria because majority of the existing antibiotics have no effect against it. So people go into hospital with some other disease, and within a few weeks of being in hospital, they pick up an infection of Acinetobacter Bomani, and it
just can't be treated.
Exactly, and it just can't be treated.
That's extraordinary.
Exactly, and then the one that's very relevant for us in developing countries, and in Nigeria particularly, is that this particular molecule, halicin, was found to have activity against Mycobacterium tuberculosis, which is tuberculosis. And as you know, Nigeria, I think, is top 4 or top 5 in tuberculosis across the world. And one of the major issues with tuberculosis now is the existence of multi-drug resistant tuberculosis strains.
So to that effect, can we say with some confidence that we are on the verge of a new age of antibiotic drug discovery through these new approaches of artificial intelligence, coupled with very careful experimental approaches, if you like?
Yes, I think we can cautiously say that.
Cautiously.
Cautiously,
Right.
and I'll tell you why. Not because of halicin, but because of the success of the model. Like I told you earlier on in the program, AI in drug discovery has been around for about a decade now. But this is groundbreaking, and this will make a lot of other people take note. The National Institute for Pharmaceutical Research and Development, which I head, has been looking in this area. You know, I don't know if I've mentioned it previously to you, that one of the first visits I made after I was appointed Director General of the National Institute was to the NCC.
Right.
And this was because we wanted to start building a database.
Right.
AI is the way forward. The National Institute for Pharmaceutical Research and Development also has been one of the agencies at the forefront of DNA identification in Africa. We've also been at the forefront of different types of medicines that, you know, focus on using data to move things forward in an efficient manner. And so this type of news will get people to wake up.
Very interesting.
Get people to wake up.
I mean, we've got a big problem in the world now of the coronavirus. It's currently causing so much concern, but it is a virus and can't be treated with antibiotics. But apparently this new AI method could potentially be used to source new antiviral drugs to combat such outbreaks. So the building blocks of many scientific discoveries have been made through this understanding.
Absolutely. AI started with self-driving cars, and
Yes,
that's
absolutely.
the good thing about this type of
Yes,
science.
It has so many
one-up it
applications.
and then use the application
Yes.
somewhere else.
Extraordinary.
That's the interesting thing. Now,
Absolutely
coronavirus, we've started hearing, we've identified three molecules that have activity against coronavirus: ritonavir, remdesivir, and our own dear chloroquine. When I heard that, it took me memories back, because I know 30 years ago, every Sunday,
Yes.
we had to swallow chloroquine
Bitter
in Nigeria.
chloroquine.
Bitter chloroquine. But who knows? With this type of discovery, because the model, like I said earlier on, we can cautiously say that we're on the verge of a new era because of the model.
Sure.
Because of the model. But halicin and its potential activity on 35 out of 36 that were tested, halicin and its potential activity against big killers like Clostridium difficile, Acinetobacter Bomani, Mycobacterium tuberculosis, and Escherichia coli. These are groundbreaking issues. So you can imagine if more people fund agencies like mine,
Yes.
partner with agencies like mine, just like the Jamil Institute, the Jamil community partnered with MIT. I mean, it wasn't just an MIT
Yes,
program.
absolutely,
It was a
yes.
partnership. So this will wake people up. We need to start partnering with people to bring machine learning to health.
So, I mean, we've got about a minute left. Is this sort of research being done at all in Nigeria and Africa? Has your agency, for instance, made it a priority to develop novel treatments for tropical diseases that afflict people in this country?
Yes, your question is in two parts. So the later part of your question, whether our agency has made it priority, tropical diseases a priority, yes, categorically.
Right.
we're going to do something before the end of the year, looking at phytomedicines that have activity on neglected tropical diseases,
Right.
because they're our diseases.
Okay.
Take it or leave it. But for the first part of your question,
Very briefly, because we're out of time.
which is whether we've started this particular model in Nigeria, the answer to that is we have the building blocks,
But you
and
haven't.
we're at the verge of starting.
Okay, Dr. Obi Adigwe, DG of the National Institute for Pharmaceutical Research in Nigeria, thank you. You're a hero.
Thank you.
You're watching the Arise interview, plenty more still ahead, including as cases of the coronavirus escalate, conservationists are calling for a permanent ban on the wildlife. We'll speak to one of them. Stay with us.